What's up, everybody? Thank you for listening in to our presentation on the TAS2R38 gene relating to the PTC lab and its connection to the 8 units of AP Biology. Today, you'll hear presentations from Kay Miles
and Sammy Wire. A quick overview of our table of contents and the lab that we did a couple days ago. On the first day of the lab, we did a PTC taste test, followed by DNA extraction. Day 2, we set up and ran our PCR amplification. And then on day 3, we saw restriction digest and we performed gel electrophoresis. Cade, who you will hear in a second, will be presenting units 1, 4, 6, and 7, as well as his slide on bioethics, followed by my slides on the remaining units and my bioethics slide.
While phenylthiocarbamide is not an actual macromolecule in and of itself, we can learn several important traits about organic chemistry just by observing its chemical properties. First, you can see a carbon-hydrogen ring involved. This is very common in biochemistry, as that's the very definition of an organic molecule. In addition, groups play a major role in defining the chemical properties of a molecule. And here, you can see in the top right that PTC has an amine group, or NH2. Next, let's cover the TAS2R38 receptor, which is a protein, but it's not a small protein, it's a polymer protein made up of multiple different amino acids. All of our complex biomolecules are made up of small, simple monomers. These monomers are simple sugars for carbs, amino acids for proteins, and nucleotides for nucleic acids. Lipids are a little too broad that we can't really give them a category for that. Unit 4 is about cellular pathways, so here we are going to cover the cellular pathway that leads to someone with the tasting alleles registering a bitter taste. Now, there are 3 types of receptors you can have in your cellular membrane: a receptor tyrosine kinase, a ligand-gained ion channel, or a G-protein coupled receptor. The receptor for the TAS2R38 gene, like about 60% of receptors in your cell membrane, is a G-protein coupled receptor. To activate this receptor, you need a signaling molecule or a ligand. It just so happens that PTC and other bitter molecules do this exact function. Now, once we have the reception occur, to actually get the response we want, we are going to need a cascade to happen, or a transduction pathway. As it turns out, inocyclyl triphosphate, or IP3, is the second messenger in this reaction. That is how people are able to register bitter taste with these receptors. Essentially, what you're doing in this experiment is copying DNA multiple times, or, for lack of a better word, replicating it. And when you do replicate DNA, you do it semi-conservatively, which means you have one strand that acts as a template, and then a DNA polymerase uses a primer to then elongate that strand. Here, the DNA polymerase is from a very heat-resistant bacteria called Thermus aquaticus, or TAC. We also use many of the pieces of biotechnology described in Unit 6. For example, we have gel electrophoresis, where you separate pieces of biological molecules based on their size using an electric current, and PCR, effectively the DNA photocopier. Now, lastly to talk about is the variations you have in the PTC gene due to SNPs. These are effectively point mutations based on tiny little changes to one nucleotide. Bitter tasting genes are a great illustration of 3 types of ways a population can evolve. First, PTC variations have been linked to, in our early hominid ancestors, balancing selection, where multiple different types of alleles and phenotypes were kept around as they provided protection against various bitter compounds. Next, there is another bitter gene called TAS2R34 that's disabled in primates, but multiple lineages have done it in different ways. This is an example of convergent evolution, where 2 completely different lineages have the same phenotype, but they did not get it from a common ancestor. Instead, they did it in 2 or more separate ways. Finally, the PTC gene has more variations in Africa than in populations outside the continent. This supports the idea of genetic drift via the founder effect, where a certain population carried out alleles that were not representative of the overall population, leading to reduced diversity. In his 2008 Dignitas Personae, Pope Benedict XVI covers 2 major issues of bioethical concern. First, he reiterates the Church's stance against eugenic practices. Embryos, he emphasizes, cannot be created to have specific phenotypic traits, nor prevented from conceiving because of their genetic makeup. In order to protect equal human dignity, gene selection in the creation of new embryos cannot occur, as it breaks this fundamental part of human morality. Next comes genetic testing, which often causes an impulse to do gene therapy. While the Pope iterates that therapy on somatic cells to cure disease is fine, doing gene editing on gametes that cause permanent changes to phenotypic traits is never permissible in the slightest.
The TAS2R38 gene that we looked at in the lab codes for a receptor protein which is located in the membrane of taste receptor cells. As we take a trip through the endomembrane system, we can see how this receptor protein is not only assembled but transported. We start in the rough ER, where ribosomes synthesize the protein, followed by the Golgi apparatus, which modifies and packages it, before vesicles transport it to the plasma membrane, where it becomes embedded and functional. The folds that these taste receptor cells have are also very important. As you can see in the bottom right picture, they are known as microvilli. Such folds increase the surface area to volume ratio, which allows more receptor proteins within the plasma membrane that improves the cell's ability to interact with its environment, like how it interacted with PTC in this lab. A major aspect of the PTC/PCR lab was restriction enzymes, which are highly specific, meaning that they only cut particular nucleotide sequences that we see in the TAS2R38 gene. This lets us focus on specific DNA patterns to identify after the DNA is cut. The use of Taq polymerase also greatly increased the efficiency of this lab by lowering the activation energy required for the reaction, which allows DNA replication to occur more often during the PCR cycles. Another notable Unit 3 connection is temperature. During PCR, temperatures reached up to 97 degrees Celsius, which denatures DNA and separates its strands. Due to the heat-tolerant nature of Taq polymerase, however, this enzyme was able to function despite the intense heat. Finally, temperature held around 37 degrees Celsius for 15 minutes provided an optimal temperature for enzymes to function efficiently. This is a major aspect of Unit 3, how enzyme activity is dependent on environmental conditions. Our PTC-PCR lab is directly connected to Unit 5, which covers heredity when we look at Mendelian inheritance of the TAS2R38 gene. This gene is inherited by and passed to offspring through alleles, each parent providing one allele. For this lab, it is important to know that the taster allele is dominant over the non-taster allele, so those able to taste PTC are at least heterozygous, as seen by the three images of a strong, mild, and non-taster to the right. After revealing each of our phenotypes by using the PTC taste strips, analyzing restriction enzymes, and running gel electrophoresis can reveal our genotypes based on the different DNA banding patterns that are produced. The relationship between dominant and recessive alleles, as well as Mendelian patterns of inheritance, are strong Unit 5 topics demonstrated in this lab. Unit 8 connects to the PTC/PCR lab by focusing on how the expression of traits can influence interactions within the environment. Because many plants often produce bitter chemicals, such as the toxins depicted in the image on the bottom right, that the PTC non-taster is eating. This can influence environmental interactions and which foods organisms choose to consume or avoid. Within a certain population, individuals can have different variations of the TAS2R38 gene, which creates variation in the ability to taste bitter compounds like PTC. Since some individuals perceive the bitterness differently from others due to their PTC tasting alleles, we can notice a difference in feeding patterns on certain vegetables or plants. Not only just by animals though, humans too see a difference in feeding patterns. While most of them were heterozygous or homozygous for the trait, we did see select individuals that were recessive for PTC tasting. In his 2015 encyclical, Laudato Si', Pope Francis discusses the new area of humanity that we have entered, especially relating to technology and science. He is quoted saying that when well directed, technoscience can produce important means of improving the quality of human life. With such power, though, comes a major risk of our human nature being exploited. He goes on to state that never has humanity had such power over itself, a bioethical concern that comes with this advance in technology is ecological consequences, which are often overlooked. Pope Francis then goes on to state how not all advancements in power are progress for a better good. This does not mean that we should halt the scientific advancements being made, we just have to regulate them. When referring to genetic engineering as a subtopic of bioengineering as a whole, we must focus on making these advancements helpful for resolving life-threatening conditions. We must make sure that homeostasis in all beings is not disrupted. We do not disrupt human integrity, nor do we hinder biodiversity.